Passive transport. This is a movement of molecules across the cell membrane without energy input from the cell. In other words, it does not require any ATP. In this diagram in the middle, you can see head and two fatty acid tails forming a bilayer, right? And the arrow is extending from left side to right side, where it is moving from areas of high concentration to areas of low concentration, where the left side is crowded of molecules and the right side isn't. In other words, it's low concentration, so it's down a concentration gradient. Facilitated transport. It is another type of passive transport which does not require any ATP, and this is a diffusion of molecules across the membrane through transport proteins. The transport protein is the purple thing in the diagram, and still the arrow is extending from left side to right side in this diagram, where it's moving from high concentration to low concentration. And it is a diffusion that is assisted by proteins such as channel or carrier proteins. Active transport. Why is it called active? It's because it does not require any energy. It moves from region of lower concentration to a region of higher concentration. This also takes place in cell membrane, and if you look at the left side, it's not crowded of molecules where the arrow starts, and the right side is crowded of molecules, so it's moving from low concentration to high concentration. And there are many types of active transport, such as endocytosis, exocytosis, sodium potassium pump, which I will address soon. So these active transports are against concentration gradient. Diffusion. It is a type of passive transport, and it is a movement of small molecules through the membrane of high area of concentration to low area of concentration, where you can see in this diagram those molecules are NaCl salts, and the left side is crowded and right side isn't, so it's moving from high to low concentration. Osmosis, another passive transport. It is a movement of water from high area of water concentration to low area of water concentration. And you can see the H2O molecules all over the place. And you can see the left side is more crowded, so it is presumably that it is moving from left to right. Hypotonic solutions. It is when there are less concentration of solute. So in this case, there are more salt inside than outside. It is giving pressure from outside to inside because based on osmosis, it's moving from low solute to high solute. So it is high likely that inside the animal cell would pop because it is keep on giving the pressure from outside to inside. Same thing for plant, which has cell wall. Since there's more salt inside, there will be turgor pressure because from outside to inside, the central vacuoles will fill with water, just like putting water on the plant, so the leaves would stand better. This was hypotonic. Isotonic solutions is when there are equal concentration of solute and solvent. So in this case, there's salt widely spread outside and inside. So there's not going to be much change because it can't move from high concentration to low concentration. So there's nothing much going to happen here. Just opposite of hypotonic solution. Hypertonic solution is when there are greater concentration of solute outside than inside. So you can see the arrow extending from inside to outside. So therefore, based on osmosis, it's moving from low solute to high solute. So therefore, the cell inside the animal will shrink because the solute will be all gone. Same thing for plant, where there's cell wall, it will lose turgor pressure because it will shrink and the membrane will shrink and is plasmolized. Facilitated diffusion. As facilitated means with help, it gets help from proteins. It is a movement of small molecules from high area to low area of concentration in this diagram left to right, where the left side is full of molecules crowded and right side isn't comparably. So it is getting aid from proteins. So the purple thing is protein channel getting aid from proteins. Sodium potassium pump. It is a movement of sodium and potassium against the concentration gradient, where it is moving from low to high concentration and using ATP. And it pumps out 3 sodiums for every 2 potassiums taken in. Pretty cool. Endocytosis is the movement of large molecules inside the cell. So in this case, a molecule just goes inside the vesicle, and the cell membrane has changed its shape, and it got its shape back. Phagocytosis is a type of endocytosis where it is eating large particles at its time. So this large molecule just goes inside the cell membrane, and then it just eats large particles at its time. So there's this phagocytosis. And finally, exocytosis is the opposite of endocytosis, where it is movement of large molecules out of the cell. So the proteins inside the vesicle tries to go out, then the cell membrane changes its shape and the vesicle is released. Then the cell membrane gets its shape back and it is released.